Hello everyone. Am I audible and visible to you? Am I audible and visible to you? Okay. So, myself, Dr. Shital Ghataliya, your faculty for Physiology. Welcome on the platform of Unacademy. Welcome to Unacademy Future Doctors. Here, we will teach you various subjects of MBBS and I am your faculty for Physiology. I did MD Physiology from MP Shah Medical College and I am educated MBBS and MD. I have teaching experience of more than 17 years and I am one faculty for integrated curriculum teaching. Let us go through some subscription details of Unacademy. With the Plus subscription, you can have access to both live as well as recorded classes. You can learn from the topmost educators of India. You can compete in live tests and quizzes. You can study on the device of your own choice. You can have access to the question bank having more than 25,000 questions in it. You can even have a printed notes available with certain subscriptions. With Iconic subscription, you can access to the best from two of the best. Unacademy and Prep Ladder. With Unacademy, you can find resources like well-structured live batches, classes, tests, and quizzes, and many more. Unacademy and Prep Ladder. With the Prep Ladder, you will find clinical and integrated essential video lectures, printing, rapid review, snapshot, and many more. So both are unique in its own way. Whatever is best suitable for you, that you can choose. Now, Unacademy came up with some special class feature. Now, what is this? The classes will be highly interactive. There will be a poll for the learners, so you can understand the content in a better way. You can even raise a hand, talk to your educator, and get your query solved. You will never miss a class because Unacademy will send you regular notifications. You can even have a lecture notes available with certain subscription. You can download it and you can even have access to the recorded sessions. You can attend the classes from any time, anywhere. As I told you, there is a highly effective question bank. There are high yield clinical questions which are based on latest exam pattern and it includes detailed explanation. These all are NEET PG toppers September 2021. Congratulations to the toppers from Unacademy. As I told you, there is a raise a hand feature. So now the sessions are highly interactive. You can be engaged in quizzes and viva and doubt solving and many more. So Unacademy Medical PG days, there are two free test rituals and plus test rituals. With the free test rituals, you can find educator curated test series, FMG grand test, study with me, YouTube test series, and many more. With plus batch test, you will find NET PG 2022 subject wise test, 2022 grand test series, and many more. So there are two batches now, Focus FMG 2022 comprehensive. Duration is 6 months, already started from 15th of December. Target NEET 2022 taste and discussion batch. Duration is 6 months, already started from 15th of December. So NEET PG subscription plus as well as Iconic subscription, both are available with various durations and different prices. You can use the promo code 10 to get the additional discount. Now study with me. Pathology test series is there. Live session is there on 14th January. Attend that. You can use the code Sheetal10 to unlock and enroll for the test. Now come to today's topic. That is functional aspects of neurons, and I will tell you the classification of nerve fibers. Now let us understand first what neuron is. functional aspects related to the neuron. Neurons are structural and functional unit. Structural and functional unit of nervous system. So, if we look at the structure of the neuron, broadly it can be divided into four parts. Nerve cell bodies, also called as soma, nerve cell body, dendrites, axon, and synapse nodes. Nerve cell body, dendrites, axon, and synaptic nodes. Let us go through the first one. Cell body is soma. Here, it is of the different shapes like round shape, pyramidal shape, etc. Cell body forms, so it loses its ability for the division. So whenever the cell is damaged, whenever the nerve fiber is damaged, it will be replaced by neuroglia only. Now there is a presence of nasal granules. Very important, nasal granules. The nasal body. These are basophilic granules. And nasal granules or nasal, they are basophilic granules. Is composed of rough endoplasmic reticulum. Rough endoplasmic reticulum. So it is important. As is right, goes without saying, the endoplasmic reticulum over which isn't granulated and granular endoplasmic reticulum. So it is important for protein synthesis. This protein is needed for maintenance, repair, production of neurotransmitter, and formation of enzyme. Right? So protein is important part of the cell body. It is formed in the nasal granules. It is formed by nasal granules, which is composed of rough endoplasmic. Important for the protein synthesis. This protein is needed for the maintenance, the repair of the neurons, production of neurotransmitter. These granules disintegrate in fine dust. Will be disintegrated in fine dust and it will finally disappear. That is called as a chromatolysis. Chromatolysis. This is it is what is chromatolysis means the disintegration of the nasal granules or nasal bodies, fine dust, which finally disappear. Okay? It happens when there is a when there is a fatigue or when there is a neuronal injury. So this is important aspect. Then there are presence of neurofibrillary. Neurofibrillary inside this cell body of neurofibrillary. That is a network of the fibrillary, the cytoplasm of the neurons. It consists of microfilaments and micro. It consists of microfilaments and microtubules. So it gives shape, you can say. So this neurofilament, this is very, very, very important. Neurofilament protein will get altered in a disease, that is Alzheimer's. Neurofilament protein gets altered in the disease that is called as a Alzheimer's disease. Okay? An altered protein causes formation of tangles, that is lesion. It forms the lesion, forms the neurofibrillary tangles. This is very, very important to remember. Neurofibrillary, that is the presence of the network in the cytoplasm of the neuron. It consists of microfilaments, microtubules. These neurofilament protein alter, that is Alzheimer's, and causes formation of the lesion that is called as a neurofibrillary tangle. Neurofibrillary tangles. Now come to the another part of the neuron, that is dendrite. Dendrite. So they are multiple, right? You can see from the diagram itself, they are multiple small branched process. Multiple and small branched. They are important as they are receptive processes of the neurons. They are receptive processes of the neuron. So it will receive the signal from the other neuron. Will receive the signal from the other neuron via their synapse, the axon terminal. So by the synapse, it will receive the signals. Another neuron. This synaptic input will cause generation of the potential. I will tell you potential, graded potential and action potential into class, special class. Synaptic input will produce the graded potential. In it is said that in dendrites, action potential is not generated. It just forms the graded potential. It means local potential change. So graded potential which can be algebraically summated on the surface of the dendrite. So it will conduct, mostly it will conduct the graded potential. This graded potential, remember this is local risk. Local risk. Okay, it can be depolarizing, it can be hyperpolar. So dendrites are the receptive processes which will receive these signals. Synaptic input will be there, right? And this input will produce the potential, local potential change. And mostly it is the graded potential. Now come to the third part. Third part, that is axon, right? So axon is a single longer process, the nerve cell. Okay. Single longer process, right? It arises from the conical extension of the cell body that is called as a axon hillock. The conical extension of the cell body which continues as axon. That conical shape is called as a axon hillock. Axon is devoid of granules, nasal granules, not there. In the cell body, it is there. In dendrite, it's there, but it is not there in the axon. The part between the axon hillock, the conical shape, and the area of the axon from where the myelin begins. This part, where the myelin sheath begins. That is called as the initial segment. Segment. This is important specifically when action potential. About action. So here too is axon hillock, conical shape extend from the cell body, that is nasal granule. Okay. The part of the axon hillock in beginning of the myelin, the axon, that is called as segment. Called as the initial segment. Now, in the axon, the cell membrane continues. Cell membrane from the cell body will continue as exoderma, and cytoplasm is called as exoplasm. And cytoplasm. The cell membrane of the axon, that is exoderma, and cytoplasm of called as the exoplasm. The axon will terminate and it will divide into multiple branches. This one divide into branches. Okay, and each end it will divide in number of the synaptic, right? So this is called as a synaptic lobes, or also called as a terminal buttons. Synaptic lobe, or also called as the terminal buttons. Now this synaptic lobes contain microvesicles, small vesicles inside it, which it contains neurotransmitters. Synaptic lobe contains microvesicles. Inside these microvesicles, neurotransmitters are present. These neurotransmitters are important whenever it receives the impulse. Because from the vesicle, neurotransmitters will be released into the left, right? So this is about the axon. Now come to the next, that is about the myelin sheath. Myelin. What is myelin sheath? Is a protein lipid complex mixture. protein lipid complex which develops on the axon, except the ending and nodes of Renvier. This one, it is not there at the end, and there are gaps. That is called as the nodes of Renvier. Right? So it is not there. This one. This is how the myelin sheath will be formed. Formation of this is the one cell and it will wrap around the axon, turn around the axon, and it will form the sheath, lipid mixture. That is, this is complete formation moving around zone, that is called as a myelinogenesis, and forms the myelin sheath. This see how many it took, right? This is the complete myelin around the axon. It is there in the myelinated nerve fibers. Myelinated nerve fibers are not there, then the myelin sheath is not there. Now, what is the function of myelin sheath? Is important. Very, 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 very important. It is having high insulating property. Whenever the impulses are propagated, at that time it will not allow the loss exchange of the ions. So exchange of the ions occur at the of Renvier. So it is having high insulating property. Now impulses confines to the individual fibers, and that how it facilitates the conduction of the from one node of Renvier to another node of Renvier. Travel like that. Okay, that is called as a saltatory conduction of impulse. So that is the importance of the myelin. In demyelinating disorder like multiple sclerosis, there will be patchy destruction of the myelin sheath. Was patchy destruction of the myelin. Now come to the difference in unmyelinated. See, the word itself says myelinated means present. Unmyelinated means not present. So in myelinated nerves, one cell membrane will coil many times around the axon and it will form myelin. In unmyelinated, axons are simply buried in the swan cell without wrapping of myelin. Right? The myelin sheath, myelinated nerves are present. Which are the sites where it could be present? All preganglionic fibers of is important from MC. All preganglionic fibers of autonomic are myelinated. Nerve fibers of somatic nervous system more than 1 micrometer in diameter. They are also myelinated. Unmyelinated, all postganglionic fibers of they are unmyelinated. Nerve fibers of the somatic nervous system less than 1 micrometer diameter. They are also unmyelinated. Now, the important difference here is in myelinated nerves, the impulse conduction very fast because the type is a saltatory conduction. This I will tell you, teach you special class. Saltatory conduction of impulse because it moves from one node of the Renvier to the other node of Renvier. Impulse conduction is fast. While in unmyelinated fibers, unmyelinated nerves, the impulse conduction is electrotonic. So the conduction is relatively slower as compared to the myelinated nerves. The conduction of the impulse is slower. So this is how it will look like: myelinated and unmyelinated nerve. You can see here the axon is simply buried in the swan cell. Okay, here the swan cells are forming the myelin. Now, so this is all about the neurons. Now come to the important which is important short note classification of the nerve short note. Okay, and from this itself can be one liner question, very very important. Some I have seen that some sort of MCQ usually come classification. So let us go through the first one, that is Erenger Gesser classification. According to it, there are three types of type A, type B, type C. Type A is myelinated, type B, type C is